One of the tenets of modern cell theory, as stipulated by Rudolf Virchow, is that every cell comes from a pre-existing cell. Cells, just like the larger systems they make up, slowly die. For this reason, it is absolutely necessary that cells divide to ensure the continuation of life.
All cells are part of a process that lasts from a cell's creation to its division called the cell cycle. The purpose of this cycle is passing on the parent cell's genome or genetic information onto offspring in order to continue life. This process of mitosis results in two identical diploid somatic cells. In humans, this means that each cell will have 46 chromosomes. Mitosis is very important for replacing dying cells and can be broken up into three major steps. The cell cycle consists of interphase, mitosis, and cytokinesis.
Interphase is the preparatory step before the cell actually divides in mitosis. Interphase has three parts, G1, S, and G2. In all three phases, the cell is producing all of the organelles and proteins needed by the daughter cells. In S phase, the cell's genetic information is doubled.
The mitotic phase of the cell cycle has four major steps. The first is prophase, in which this chromatin of the nucleus becomes more condensed into chromosomes consisting of two sister chromatids. The mitotic spindle forms as microtubules push the centrosomes towards opposite ends of the cell. The nucleoli disappear. The nuclear envelope fragments and kinetic cores attach to the centromeres of the sister chromatids. Some of the microtubules connected to the centrosomes attach to the kinetic cores, and others interact with microtubules stemming from the opposite centrosome.
In the next phase, metaphase, the chromosomes line up along the metaphase plate. This plate is a plane between the centrosomes. Each centrosome has one microtubule attached to a sister chromatid for each chromosome.
In anaphase, the bonds between sister chromatids are broken and kinetic core microtubules pull the chromatids toward the centrosomes. Meanwhile, the non-kinetic core microtubules from opposing centrosomes push each other apart.
In telophase, the chromatids finish moving towards the opposite sides of the cell. The nuclear envelope begins to reform, the nucleoli reappear, and the chromatids become less condensed.
The last phase of the cell cycle, cytokinesis, marks the division of the cell into two separate cells and often occurs alongside telophase. During cytokinesis, microtubules create a cleavage furrow that pinches the cell in the middle as the cytoplasm and organelles are evenly distributed amongst the two cells. In plants, there is no cleavage furrow. A cell plate is constructed between the two future daughter cells and causes a separation into two separate cells.
The other type of cell division is meiosis. This division creates gametes, which are haploid cells, meaning that they have half the number of chromosomes as somatic cells. In the case of humans, that number is 23. Meiosis and gametes are extremely important for sexual reproduction and genetic variation. Meiosis produces four genetically unique daughter cells by undergoing prophase, metaphase, anaphase, and telophase, twice after going through interphase.
Prophase 1, the newly replicated homologous chromosomes pair with each other via synapsis to form tetrads. In synapsis, homologs exchange DNA with each other in order to create unique chromosomes. Spots where this crossing over occurs are chiasmata. Like mitosis, the centrosomes move towards opposite sides of the cell, the nuclear envelope deteriorates, and tetrads move towards the metaphase plate. In metaphase 1, tetrads line up on the metaphase plate. Both of the homologs in the tetrad attach to a kinetic core microtubule.
In anaphase 1, the bond between the two homologous chromosomes in the tetrad is broken. The centrosomes pull one of each homologous chromosome away from the center of the cell. In telophase 1, nuclear envelopes reform as chromosomes become less condensed. Two nuclei with a haploid set of unique chromosomes form. At the same time, cytokinesis occurs, just like in mitosis.
After meiosis 1, meiosis 2 occurs. Meiosis 2 is the same as meiosis 1, except there is no interphase before meiosis 2, no crossing over, and the process of anaphase separates the sister chromatids of each chromosome rather than separating the homologous chromosomes from each other.
Prophase 2 is similar to prophase in mitosis. Centrosomes move towards opposite sides of the cell, and sister chromatids move towards the metaphase 2 plate. The chromosomes line up along the metaphase plate and microtubules attach to the kinetic cores of the non-identical sister chromatids.
In anaphase 2, the protein binding the sister chromatids is broken and the microtubules pull sister chromatids to the centrosomes. In telophase 2 and cytokinesis, the nuclei forms, chromosomes decondense and the cleavage furrow forms. The result is four non-identical cells consisting of 23 chromosomes each.
Both mitosis and meiosis require replication of DNA in interphase before division. Mitosis divides a cell into two identical diploid cells, while meiosis divides one parent cell into four unique haploid cells. Meiosis produces these cells with synapsis, where non-sister chromatids cross over genetic information. Mitosis allows for growth, repair, and asexual reproduction in organisms, while the process of meiosis is important for genetic variation and sexual reproduction.